Hello, good morning, students. In the last lecture, we had studied about protein and the bonds present in protein, that is, a peptide bond or amide bond. Due to presence of this C double bond ONH bond, that is a peptide bond, many peptide linkages are present in protein. Therefore, proteins are also called as a polypeptides. The proteins are responsible for variety of functions in organism. For that purpose, we will study the structure of protein. This protein of hair, muscle, skin give shape to the structure of these parts, while enzymes are the proteins which catalyze physiological reactions. So it is a very important role of protein. These proteins present in the hair, muscle and the skin. They give shape to the structure, and the enzymes, these are the proteins which catalyzes the physiological processes in our body. The deserved function of protein can understood by studying the four level structure of protein, namely this primary structure, secondary structure, tertiary structure and quaternary structure of protein. We will study all these structures in detail. First, primary structure of protein. So primary structure of protein that shows the sequence of the constituent amino acid residues linked by peptide bond, meaning is how these amino acids are linked together in a protein with the help of peptide bond, that is explained in primary structure. Any change in the sequence of amino acid residue results in a different protein. So if there is a change in the sequence of amino acid, then the protein will be changed. So the structure of protein, therefore it is very important. The primary structure of protein represents by writing three-letter symbol of amino acid residue as per their sequence in the concerned protein. So how these amino acids are written in the primary structure, that is shown with here as an example. Here GLY, TYR, GLY again, ALA, PHE, VAL. So these are the three-letter symbols of the amino acids, and they are linked together in this way. So this GLY, that is a glycine, and TYR, tyrosine, this is a dipeptide, that is the two amino acids are linked together by a peptide bond. Similarly, here the example of three amino acids linked together, it is given in a tripeptide. So it is GLY means glycine, ALA means alanine, and PHE means phenylalanine. So this is a tripeptide, and here VAL means valine is a alpha amino acid. So this is the sequence of alpha amino acids that is shown with the help of three-letter symbol in a primary structure. The symbols are separated by a dash. According to convention, N-terminal amino acid is written to the left and C-terminal amino acid is written to the right side or the right end. This is shown in this structure. So here this is NHCHR, C double bond ONH. This is N-terminal because NH group is present at the left end, called as N-terminal. And here again the groups bonded are CHR, CONH, CHR, CONH, again CHR, CO. So here at the right end there is a carbonyl group and therefore it is called as a carbon terminal. So this is a representation by structural formula of protein. Now, with the help of three-letter symbol, we can represent protein like this. ALA means alanine, GLY means glycine, SER means serine, then TYR means tyrosine, then GLY is again glycine, GLY is again glycine, like this. Alanine is an N-terminal and lysine is C-terminal. So this is represented with amino acid symbols. So that is about the N-terminal and the C-terminal in the protein, and that explains the sequence of alpha amino acids in the given protein, and this structure is called as a primary structure of protein. Now we will study secondary structure of protein. So what is secondary structure of protein? Here the three-dimensional arrangement of localized regions of protein chain. Here 3D arrangement of localized regions of protein chain is called as a secondary structure. So what is a secondary structure of protein? It is a 3D arrangement of localized regions of protein chain. It's called secondary structure. Here the hydrogen bonding is present between NH group of protein of one amide and C double bond O oxygen of another gives rise to secondary structure. This we will study in this diagram. Before that, the two types of secondary structure commonly found in protein, these are alpha helix and beta pleated structure. So how these alpha helix and beta pleated structures are there? So this is shown in the backbone of alpha helix. See here, this is N terminal and this is C terminal. Here the groups present here are NHCHRCO, 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 again NHCHRCO. This is a linkage of alpha amino acids present in the protein. Here the CO, that is the carbonyl group of one alpha amino acid, that forms this dotted bond called as a hydrogen bond with NH group of other alpha amino acid. Similarly, this is CO group of one alpha amino acid that forms hydrogen bonding with NH group of other amino acid. So in this way, this CONH bonding is present, and this is alpha helix structure of protein. So what is this alpha helix structure that we will study in detail now? This alpha helix forms when a polypeptide chain twists into right-handed or clockwise spiral. So this is right-handed twist is there. So there are chain twist into right-handed or clockwise spiral. So this is one right hand, this is also right-handed, right-handed twist is there, and that gives the alpha helix type of structure. Then some characteristic features of alpha helical structure of protein are: each turn of alpha helix has 3.6 amino acids. So here this one alpha amino acid, second alpha amino acid, third alpha amino acid, and this is half amino acid. So it is a 3.6 residues of alpha amino acids are present in the each turn of this alpha helix. That is one of the characteristic. Then the carbonyl group of one alpha amino acid is hydrogen bonded with NH group of fourth amino acid along the chain. Now we will see how this CO group of one alpha amino acid is bonded with fourth uh, NH group of alpha amino acid. So see, this is NH CHR CO. This is one alpha amino acid. NH CHR CO. This is second alpha amino acid. Then NH CHR CO. So it is first, then second, then it is third, and now it is fourth. So this CO group of this fourth amino acid that get bonded with NH group of next fourth alpha amino acid. See, it is how it. So it is first, then it is second, then it is third, and this is the fourth, and this is the NH group of fourth alpha amino acid. So that is linked in alpha helical structure. So that is the second characteristic of alpha helical structure of protein. The third characteristic of alpha helical structure is hydrogen bonds are parallel to axis of helix, while R groups are extended outwards from the helix core. So hydrogen bonds are parallel first. So see this dotted bond, this is dotted bond, that is called as a hydrogen bond. So these bonds are parallel. Similarly, these two dotted bonds, they are also parallel. These two are also parallel. These two dotted bonds, that is hydrogen bonds, are parallel. And this R groups extended towards outward from the helix core. So see this R group here outside this core. This is R group outside. If it is present to the below of this line, then it is called as a inside, but it is outside this line. Hence, that is outer core. This is also outer core. Outer core R group is present. So these are the characteristics. Which characteristics we had studied? So each turn of the alpha helix has a 3.6 alpha amino acid. Then CO group of one alpha amino acid is hydrogen bonded with NH group of fourth amino acid in the chain. Then hydrogen bonds are parallel and R groups extend outward from the helix core. Then myosin is a muscle and alpha keratin is hair, that is a proteins which almost entire alpha helical secondary structure. So which protein have alpha helical structure? Here two examples are given. One is myosin, the protein present in muscle, and alpha keratin, the protein present in hair. They have alpha helical structure. Then this beta plated structure, we had studied now alpha helical structure. Now it is a beta plated structure. The secondary structure is called beta plated sheet when two or more polypeptide chains are these two or more polypeptide chains are there, it is called as a strands, and they line up side by side. So see this violet color structure given in this figure. So it is a one chain of polypeptide chain, and this is other polypeptide chain. If they are bonded with hydrogen bonds, then this type of structure is called as a beta pleated sheet. Here the two or more polypeptide chains are called strands. They are line up side by side. This is shown in the figure 14.20. So here these are the sheets are shown, and here actual structure, that is NH CHRCO, NH CHRCO, NH CHRCO. This is one polypeptide chain. It is hydrogen bonded with other polypeptide chain, that is NH bond of one polypeptide chain get hydrogen bonded with CO bond of other polypeptide chain. Like this, there are many hydrogen bonds between polypeptide chain, and this type of structure is called as a beta pleated shape. Similar to characteristics of alpha helical structure, we will study this characteristics of beta pleated sheet. So these characteristics are: first is C double bond O and NH bonds lie in the planes of sheet. So these bonds CONH, CONH, these bonds are lie, they are present in the planes of the sheet. So in the same plane they are observed, not below or not above. Then hydrogen bonding occurs between NH and CO groups of nearby amino acids. This hydrogen bonding is present, we had studied, between NH group and CO group. They are of nearby amino acid residues in the neighboring chain. They are not for the fourth or third amino acid, but they are to the nearby groups in the polypeptide chain. Then third characteristic is R groups are oriented above and below the plane of the sheet. So this one R group is above, other R group is below, then next R group is above, then next R group is below. Like this here in the one sheet, one polypeptide chain, this R groups are one above and one below. They are placed alternately, unlike to this alpha helical structure, and these are the characteristics of beta pleated structure. Then this beta pleated sheet arrangement is favored by amino acid with small r groups. Remember this well. This, if R group in the alpha amino acid is a small, then it prefers the beta pleated structure. The most proteins have regions of alpha helical and beta pleated sheet in addition to other random regions that cannot be characterized by either of these secondary structure. For example, the spider dragline silk protein is a strong due to beta pleated sheet region. It elastic due to the alpha helic, strong due to beta plated structure, and elastic due to alpha helical regions in it. So that is one very good example. Then next type of structure of protein is a tertiary structure of protein. The three-dimensional shape of protein is adopted by entire polypeptide chain of protein. This three-dimensional shape is adopted by entire polypeptide chain of protein, and this is called as a tertiary structure of protein. This results in the folding of chain in a particular manner that the structure is itself stabilized and also has an attractive interaction with aqueous environment of the cell. The globular and the fibrous protein, these are the two different types of proteins. They represent the two molecular shapes which results in the tertiary structure. So which are the examples of the tertiary structures of protein? This globular protein and the fibrous protein. The force that stabilizes a particular tertiary structure is again hydrogen bonding and dipole-dipole attraction. So which bonding or which force stabilizes the tertiary structure? It is a hydrogen bonding and dipole-dipole attraction due to polar bonds in the side chain. Then other type of force is electrostatic attraction. Why electrostatic force of attraction? It is due to ionic groups like COO minus and NH3 plus in the side chain, and also one more type of force that is called as a London force or the London dispersion force. Finally, disulfide bonds formed by the oxidation of nearby SH group, that is in cysteine residue, all are the covalent bonds which stabilize the tertiary structure. Here in the figure 14.21, tertiary structure of protein is given. This is a strip which is folded like this. It is a polypeptide chain having hydrogen bond, having alpha helical structure, having beta plated structure, having disulfide bond, having electrostatic attraction, London force of dispersion force, hydrogen bonding. All these types of attractive force are present in the tertiary structure. Then the fourth type of structure present in the protein is a quaternary structure of protein. When two or more polypeptide chains are folded with tertiary structures, they come together into one protein complex. This tertiary structure, they come together into the protein complex and which results in the quaternary structure of protein. So what is a quaternary structure? When two or more polypeptide chains folded with tertiary structure and they come together, they forms a structure of protein, complex protein, that is called as a quaternary structure. Thus, each individual polypeptide chain is called as a subunit of the overall protein. For example, hemoglobin consists of 4 subunits called as a heme, held together by intermolecular force in the compact three-dimensional shape. So we will study this quaternary structure. This hemoglobin can do its function of oxygen transport only when all the 4 subunits are together, and that is summarized in the 4 levels of the protein structure, that is alpha protein, alpha amino acid, beta pleated sheet, this is a beta pleated, again alpha helix. So this is a primary structure that shows the sequence of alpha amino acids. So these are joined together to form the primary structure. In the secondary structure, this is a polypeptide chain called as the alpha helix. And these are the polypeptide chains. These are called as a beta pleated sheet. This we had studied all these structures. Then tertiary structure, here the beta plated and alpha helix, they are bonded together, and finally all these types of bonds along with this hydrogen bonding, dipole dipole attraction, electrostatic attraction, London dispersion force and the disulfide bond, these are formed in the quaternary structure. These are the multiple units of tertiary structure, and that is called as a quaternary structure. So that is about the quaternary structure, and we had studied the structure of protein in 4 levels: primary structure, secondary structure, tertiary structure, and quaternary structure. Thank you.